Alright, so today we're going to begin Unit 5, and we're looking at this unit. It's protein synthesis, and then we'll get into mutations. Today we're going to look at a new piece of genetic material. We've talked about DNA, the structure of DNA, how it's built, the nitrogen bases that pair together. But now we're going to look at RNA. Okay. To understand RNA, we have to look at the differences between the two. All right, trying to get my face off the screen for a second. So DNA, RNA, similar. The names are a little bit different. Remember deoxyribonucleic acid, that deoxyribo part described the sugar. Well, now that's changing. We don't have deoxyribose anymore. Now we just have ribose. That's why we get RNA. All right. Second thing, RNA is single-stranded, one half of it. DNA is the two strands put together in the ladder. Now we just have half a ladder. That's what RNA is. I already said the sugars, deoxyribose versus ribose. That's one of our main differences here, is the sugar that's in there. All right, so we have that sugar difference. Right here we have deoxyribose for DNA. We have ribose for RNA. That's a big difference. Okay. Also, with our base pairing, remember we have our apples and trees, our car in the garage. Well, now we've changed. There's a little bit different. Instead of having T, because RNA has no T. Okay. So for RNA over here, we put no T. There's no T's in RNA. We replace that with the U. So A now, apples, go in the U. Find something to understand and remember that, but that's what it is. A's go with U's in RNA. All right, so anytime you see a U, you know it's RNA. Anytime you see a single strand, you know it's RNA. Okay? Now, why do we need to do this? The reason why is because we need a copy of that code to be able to do some other things. That code of DNA can only exist inside the nucleus. Only in the nucleus. All right, that's a little funky circle there. But it doesn't leave it. You know, it's kind of like it's safe zone. If I have $1 million, I'm not just going to leave it laying on the front porch. I'm going to lock it up. It's going to be in the bank, or it's going to be in a safe. It's going to be locked up somewhere because it's important. It's valuable. Same for DNA. DNA, if it gets damaged, cells, you get all these weird things happening inside cells. We keep that locked up in the nucleus. So if we need a copy of that to go somewhere, that's what RNA is. All right? RNA can be found in the nucleus. All right? It can be found floating around in the cytoplasm, and then it will be at the ribosome, which we'll see why coming up in this unit in the next few days. Okay? So some pictures of it here. We have RNA versus DNA. Here's our DNA. Of course, it looks like that normal twisted double helix shape that we talked about. We've got A's, G's, C's, and T's. My face is covering up the C there. There we go. A, G, C, T. All right. RNA is just one half of it. It's one rung of the ladder. All right. And now we still have C, we still have G, we still have A, but now we have this U. It's just a little bit different. It's not much different. The only thing that's missing here is we're missing one carbon out here. It's almost the exact same, but that does make a huge difference. Okay. So we have three types of RNA. We have mRNA. M means messenger. Okay. M for messenger. That that RNA, messenger RNA, is going to take the code from the nucleus to the ribosome. All right, so you figure you got your nucleus here. We've got our DNA inside. We've got our weird shaped ribosome here. I need this code to get to the ribosome. All right, I need to know what that code is. So instead of the DNA coming out, we make a copy of RNA, which is a single strand. It can leave the nucleus and go to the ribosome. All right, that's messenger RNA. It's taking the message, taking the code to the RNA. Okay. Inside the RNA, we have ribosomal RNA. That's rRNA. This is the RNA that is used when we're making proteins. Right? That's what ribosomes do. We talked about that a long time ago. Ribosomes make proteins. Well, how they make proteins is by reading that RNA and building those proteins that need to be built. All right. Now, we also have transfer RNA, tRNA. That tRNA is the one that brings in the amino acids to build the protein. All right. So right up here on this drawing here, each of these little green dots is an amino acid. There's an amino acid. There's one, there's one, there's one, there's one. The tRNA, which is what this little guy is right here, he brings in the amino acid to build the protein. Okay. Now, if you look at it upside down, it kind of resembles a T. tRNA. Okay. That's what T are, transfer. They're transferring the amino acids to the ribosome to build the protein. All right. So those are our three forms of RNA. You can go in now and you're going to do the RNA versus DNA comparison worksheet, and that's going to be your assignment and your attendance for today.